Okay, reshooting this video because the first time the text was too grainy to see. Welcome back to Knowing West Corner. I wanted to do a video for this key instead of a normal paper key because I really want to explain the kinds of thought processes involved in doing these kinds of problems. Experimental setup problems require more than rote memorization, and you really have to understand the process behind what is going on in each experiment. Furthermore, there's usually a specific concept that's being tested in each problem, and if you can identify the concept being tested, you can very more easily see what you're supposed to do. I want to preface this by mentioning that there's an organic chemistry course taught by a certain professor, and that professor's Culper reviews say that the exams require magical chemistry revelations. I've seen those exams. They do require magical chemistry revelations. These biology exams do not require magical biology revelations. You don't have to suddenly be struck by magic and divine intervention. If you understand the process behind what is going on, and you understand what we're testing, then you can very clearly see the pathway that you need to go by. I'm going to skip question 1, because question 1 relies on some information from question 2. The background here is that we have some gene that we want to put into yeast via plasmid vectors. So I'm going to go to question 2. We're splitting this gene across 8 plasmids. And I want to know what's on each plasmid. Plasmid 1 is different than plasmid 7, 1, 2 through 8. That's a pretty big hint. So we're going to start by drawing out a general drawing of a bacterial operon system. You got a promoter, you need an operator, and then you have various genes. Therefore, the beginning of this sequence is a bit different from the rest. Your promoter and your operator have to be on your first plasmid, and then the other 7 plasmids can have genes on them. This question, if I were to ask the question, what am I testing? This is testing, do you know how a piece of DNA looks? What are the things on a piece of DNA? However, this is just a more sophisticated version of asking the same question. What has to be at the beginning? What's later on at the end? Oh, nuts, that's question 1. Okay, this is question 1, my bad. We'll revisit this in a moment. There's one thing we need to add, but we're going to do question 2 so we can know what that thing is. Question 2 gave you a setup in which you were working with plasmid 1, and you injected a sample of plasmid into liquid yeast culture. In this yeast culture, you'll have a couple of yeast cells. As you know, incorporation of plasmids is reasonably rare. Let's say 1 in 4 cells receives a plasmid. What I did next was that I took all the cells in the sample and I plated them on agar. Agar is non-selected media. It's pretty much jello. Anything can grow on agar. So therefore, all of the cells that were in this culture can produce colonies. Is this more or fewer colonies than we would want? What we want are only colonies of the cells that had plasmid in them. Therefore, the plasmid has to have a resistance gene on it. And we have to plate on selected media. For example, you could use that would reduce the colonies we have to just the ones that have the plasmid in them. Therefore, when we go back to question 1, we're also going to have to put genes for resistance. Although no one put it, so I decided not to count off. If I were to say, what is question 2 testing? Question 2's big idea is, do you know why each step of genetic engineering happens? Sure, you played it. Why? You put plasmids in. Why? Why am I using selective media? And if you forget to do something, what happens? What are the steps? Don't just memorize them, but know why they're done. Question 3 was a more sophisticated version of a vocabulary question. I wanted to know how do I test the DNA that I've got in the right sequence? The hint I gave you said, how do I know how to replicate a target sequence? We know how to do that via PCR. PCR works by, you have two strands, you use each of them, and you put on each strand a primer. If the primers combine properly, you have the right strand. If you don't have the right strand, the primers won't bind properly. Assuming that PCR works, you get a lot of DNA that you get to use. If PCR doesn't work, you know that you don't have the right sequence. Another method you could use, which was mentioned in the morning lecture that day, so I didn't expect anyone to use it, is using restriction fragment polymorphisms. Essentially, a restriction fragment has to sit at a specific point. If it cuts at that point, and you know how many fragments should be made from using a specific restriction fragment using a specific restriction enzyme, then you know that you have the right DNA. If it doesn't cut the way you want, you have the wrong DNA. And the last thing you could use is you could use a probe. Double-stranded DNA, denature, stick a probe on. I'm going to make this probe wiggly to represent that it's fluorescent. If the probe sticks to it and it glows, you know that your DNA was the right DNA. If it doesn't glow, not the right DNA. So what is this question testing you? This question is testing you, what are the methods of, what is the definition of either PCR or RIPLIPS or probing? Question 4. I quite like question 4. Because I think it's exciting. Cancer is a very interesting disease. Here's my typical bacterial piece of DNA. I have my promoter, I have my operator, I have my genes. For every operator, you have a repressor protein. And that repressor protein has some effector. And effectors can either be induced inducers or co-repressors? I think we've discussed this ad nauseum. So my question was, how do you make something constitutive? Question of the day. Define constitutive. Constitutive means that the protein is always being made. The gene is always on. How do I change the repressor protein so that the gene is always on? Well, that means that I cannot have any binding with the repressor protein and the DNA. That's one way to do it. If the repressor protein won't bind the DNA, DNA is on, machine's working overdrive, cancer. Another way to approach this is, what if we messed on the other side of the repressor protein? What if we somehow changed the way the effector works with the repressor? So we have to look at the inducer case and the co-repressor case. Either way, our goal is to have it always on. The gene, I mean. The gene is always making protein. So in the case of the inducer, you want the inducer to always bind the repressor protein, which would knock this into the on state. Inducer attach, gene on. If the co-repressor is something like a brake, you want the brake to be broken. Not attach, gene on. Those are the three ways you can cause cancer in this kind of model. Again, the question is, what is constitutive? Question 5 and 6 are virus and bacteria. And I hope it was relatively obvious that you were supposed to compare and contrast bacteria. Question of the day, compare and contrast bacterial and viral reproduction. We know that bacteria reproduce by binary fission. You need a fully functional bacterium cell, which will split, and produce quite a bit of protein. This has to be a full, living, working, protein-making cell. Viruses, in contrast, they've got their protein coat and their nucleic acid. It can be DNA or RNA. And you know that viruses will just make a boatload of stuff. So a lot of protein, nucleic acid, and that will just fall together into new viruses. You don't need a living, breathing viroid in order to make new viruses. You can assemble it from the parts. Bacteria cannot be assembled from parts. So therefore, if I have syringes of nucleic acid and protein, these are the parts. If you inject someone with bacteria parts, you don't expect anything to happen. They can't reproduce that way. But viruses come together from parts. Therefore, inject someone with parts, they'll get the virus, they'll get sick. Question of the day, compare and contrast the reproduction methods. The very last question was a freebie because I defined them for you. Question of the day was define lysis and lysogeny. Maschwitz didn't use the actual terms in lecture, but they are words that you would have known had you done the problem sets. Do the problem sets. They're important. Seriously, do the problem sets. You will not pass this class if you don't do the problem sets. So, lysis. What is lysis? Host. Virus comes in. Virus makes a lot of parts. Host explodes. Releases all the parts. Parts come together, making virus. What is lysogeny? Host. Virus comes in, makes a little bit of part, makes a little bit of parts. When the cell replicates, it'll make just enough parts to have a little bit of virus in every daughter cell. And that will keep happening. Oh, this is so much hard work. Later on, these will lyse and release parts, but the main idea with the lysogenic cycle is that the virus will reproduce with its host. This makes it appear dormant because there's no explosion, no explosive death. Explosive death means you have to make a lot of parts very, very fast. Lysogeny means that you have to make very few parts rather slowly. Therefore, fast. That's symptoms which fade. That says to me the dictionary definition of lysis would probably be reovirus. Oops. There goes my chalk. Sorry, backboard. In contrast, something that's dormant for a really long time, say herpes, is lysogenic. So I just want to repeat one more time, you need to keep on top of your stuff. But for every kind of challenge or logic question that you receive, first you need to recognize what process is involved and have an understanding of how that process works. So you need to understand, for example, bacteria and viral replication, because that's not going to be handed to you. Why do those happen the way they do? And then second, you need to know, what am I testing you on? There is some simple or relatively simple topic that each question is based in. And that goes for the exam as well. We went from something big and scary to check that I've incorporated the right DNA sequence. Which laboratory method should I use? Think of forensics. And the simpler version of that question was define PCR, define usage of RIPLIPs, define probing. Most of you should be able to spit out that definition at a drop of a hat. So recognize what the question is asking you, and then be able to answer to it. Or bacterial parts are injected into someone. Do they get sick? Viral parts are injected into someone. Do they get sick? The question being asked was, compare and contrast viral and bacterial replication. Are they the same? Are they different? And how do they work differently if they are different? And what are the ramifications of that? Anyway, if you have any questions on this material, feel free to shoot me an email. And with that, I wish you best of luck on Thursday. I'll say this on Tuesday as well. I believe in you. You're going to be fine. Study hard.